If providing antibiotic resistance to particular gene, it means that that gene, suppose the that particular gene is providing antibiotic resistance against ampicillin. So we mark it as ampicillin R, and then the transformed cell would be grown in a medium that is containing ampicillin. So the transformants which would be having the gene for ampicillin resistant, they would survive in that medium, and the rest would be, let's say, they would be rejected and they would die, and they would not be, obviously you won't be choosing those without the antibiotic resistance for cloning purpose. So this is how we explain the marker gene. Yet there is another, there are certain other, there are many examples such as tetracycline resistance. TET, we write it as TETR. So that is providing resistance against tetracycline, that tetracycline antibiotic. Similarly, ampicillin R, ampicillin for against the ampicillin. Then there could be a certain gene which is coding for a specific enzyme. Right? So there could be certain gene that is coding for certain enzyme. For example, the gene is coding for beta galactosidase. This is another selectable marker gene which is produced by LAC-Z gene, okay, of lac-operon, and it could be this particular, because LAC-Z gene, it is making the specific enzyme beta galactosidase, and it could be detected by color reaction. Some vectors can have more than one selectable marker, such as, for example, there is one example for a vector is, that is plasmid P ACY 177, right? This has the genes, two type of genes, marker genes, both for ampicillin and kanamycin resistant gene. Similarly, there are other vectors which are called as shuttle vectors. Shuttle vectors, when I say that, what are shuttle vectors? Shuttle vectors. Shuttle vectors are the vectors which shuttle between two type of organisms, two type of hosts. They have two type of host cells, means from bacteria to, let's say, yeast cell, and so on and so forth. Such type of shuttle vectors, they operate in two different organisms, so hence they have two different type of selectable marker genes. One example we can take for this is the yeast episomal plasmid. It has two genes, one for ampicillin resistant and second one is LEU2 gene, for which will synthesize the amino acid leucine. So these are different type of selectable marker genes. There are another marker genes which are also called as lethal genes. So what are lethal genes? Lethal genes are the ones that favor the selection of only those host cells which have chimeric DNA. Means the one which has been transformed, or you can call it they are transformants. Means they are having, the vector is having the insert, the desirable DNA which we have inserted. If it is there, so these genes would only promote the multiplication of the chimeric DNA. Chimeric DNA is this DNA, and without inserts, those cells would be killed. So that way is the lethal genes. They are also serving as the selective markers. One example of this is Barnase. So example of lethal gene is Barnase, that is a type of ribonuclease secreted by the bacterium. The bacterium is Bacillus amyloliquefaciens. Amyloliquefaciens by a selectable marker gene present on plasmid PMT413. Right? Again, I repeat, Barnase is secreted by this particular bacterium and Bacillus amylophaciens and by a selectable marker gene present on the plasmid PMT413. So these are selectable markers. Yet there are another type of another feature this should have, that is a plasmid should have cloning vector should have, that is reporter gene. So what is a reporter gene? Reporter gene, it is that segment of DNA which facilitates the screening of the successful clones. Right? That would, in a way, as the name is suggesting, it is a reporter gene which reports about something that it would help in screening of successful clones. Now, for example for this is LAC-Z alpha fragment, and its product would give a color reaction, bluish white color reaction. There are another good features about the ideal vector is that it should be smaller in size, because if the vector is long, it would break down during purification and it is tough to manipulate those larger size. So another feature is it should have a smaller size, right? Means its molecular weight should be low molecular weight. So these are the features of a ideal vector. Then we come to the next point. Next is types of cloning vector. So types of amongst the cloning types of cloning vectors, we'll be discussing only the plasmids for now. What are plasmids? Plasmids are circular, double-stranded, extrachromosomal DNAs which are present in the cytoplasm in a number of bacteria types, in some archaebacteria as well as in eukaryotes. So they are present in bacteria, they are present in archaebacteria, they are present in eukaryotes also, such as yeast. So their role is they are self-replicating. These can replicate on their own and they have origin of replication or ori. They have also replication control system. They have certain genes for specific functions and they are most widely used cloning factors. Types of plasmids. On the basis of number per host genome, plasmids can be either single or they can be single copy plasmids which are maintained at one plasmid per host genome, and they can be multiple, means many copies of plasmid per host cell. They can be present in many numbers. On the basis of specific genes present, plasmids are of three types. What are these three types? One is F plasmids. Okay. Then another is R plasmids. Then is Col plasmids. What are F plasmids? F stands for fertility. F plasmids, they have TRA gene for sexuality and they assign maleness to the bacterium. So they are, such bacteria are called as F positive, right? Then resistance, or R stands for resistance are plasmids. They were genes for antibiotic resistance. For example, we just talked about ampicillin resistance. We also talked about tetracycline resistance gene. Then third one is the cold plasmids. Plasmids means which encode for colcine proteins that kill the sensitive strains of E. coli. So these are different types. On the basis of their size, plasmids can be, depending upon the size, they can be mini plasmids, they can be mega plasmids. So depending upon size, as per the size, they are mini plasmids, they can be mega plasmids. Mini plasmids means they have 1 kilo base pairs, and bigger than that, bigger than 1 kilo base pairs, many kilo base pairs, they are called as mega plasmids. And according to their role in sexual reproduction, plasmids can be conjugative, okay, which promote the conjugation, promote the chances of conjugation between two different bacterial cells. So they are called as conjugative plasmids, and they can, if they are not promoting the conjugation amongst the between the two different strains of the bacteria, so they would be called as non-conjugative plasmids. Characteristics of an ideal plasmid. What are the characteristics? Although we have discussed in generally the characteristics of the vectors, they should have low molecular weight, right? Because they are easily manipulated and they do not break down while purification. They should have one or more cloning sites, means polylinker sites. It should be polylinker. They should have one or two or more ori, that is region of replication or site of initiation. They should have selectable markers, selectable genes or marker genes they should have. So certain, we'll discuss certain cloning vectors, certain plasmids which are used as the cloning vectors. So here there are mainly, one is PBR 322. It is an artificial plasmid and it is prepared by from RR1 strain of E. coli, and it bears, you can see here, it bears two sites for antibiotic resistance. One is it bears it bears the resistant gene, that is ampicillin resistant gene, and second is PBR. It is bearing ampicillin resistant gene and second tetracycline resistant gene. Second, it has an ori, you can see, that is E. coli ori, and there is one coding site which for the proteins that are involved in replication of plasmids. So these are, that is PBR 322. There is another one that is called as PUC 1919 plasmid. It bears polylinker sites, that is MCS, or an ori for E. coli. Here is ori for E. coli, antibiotic resistant gene, that is for ampicillin. Then there is one gene which is LAC-Z gene that would control the synthesis of beta galactosidase enzyme. And we have discussed its role. Then another is yeast episomal plasmid. YEP in short we are writing it. It is a type of shuttle vector and it can exist in both yeast as well as it can exist in E. coli bacterium. Hence, because it is shuttling between the two organisms, therefore it is called as the shuttle vector. And it has two type of ori. That means it would, because it is shuttling between E. coli and it is also shuttling, it can replicate within E. coli, it can be in the yeast. Therefore, it should have region of replication or initiation. It should have for E. coli as well as for yeast cell. Similarly, it has ampicillin resistant gene. It has LUC, two gene that would code for amino acid leucine. So these are the different type of plasmids. There is another plasmid which is called as TI plasmid. TI plasmid. So TI plasmid, it is found in Agrobacterium tubi. It is found in Agrobacterium tumor faciens. Agrobacterium tumor faciens. So it is found in this organism that I am talking about, TI plasmid. So TI plasmid, it is a Agrobacterium tumor faciens. It is a bacterium which resides in soil, and it causes gall tumors in dicot plants. It has tDNA. Here you can see it has tDNA, which increases the synthesis of growth regulators such as auxins and gibberellins, and it transforms the normal cell into a cancerous cell or gall tumor. So these are different type of plasmids as vectors. What are the limitations? Limitations of the plasmids is that they are able to carry only small sized foreign DNA. That means that is their limitation. Only 1 to 20 kilo base pairs. This much insert they can carry. So this is their disadvantage. So that is all about the plasmid vector.